I wanted to come to you really quickly to actually help you out. This semester, our first lecture exam is happening. So, what I thought I'd do is go through quickly the study guide. I was just going to highlight some of the questions that we went through. Um, Instead of working from front to back, I think I'm going to work from back to front since that's the most recent to me. If this helps you at all, I'm happy to share it and continue to do it throughout the semester. But I thought I would share my current thoughts on what would be the first exam. Just pretty much going to make a separate video on that. I'm going to make that tomorrow. But in terms of the questions, I've already highlighted the ones I think she's going to do or select. So I'll do that tomorrow. Okay, so I'm going to work backwards. This is chapter 5, and I'm going to start here. So basically, the first one says, explain the purpose of adding nitrates to meats. So the reason why nitrates are added to meats is because it helps prevent endospore germination. And so we remember that endospores are actually a resistant, one of the most resistant forms of the cell, and that is resistant to heat, desiccation, kind of rapid pH changes, any kind of harsh environment that you can imagine. The endospore can survive. So in order to avoid some kind of germination, in order to avoid germination, we're going to ensure that we add nitrates to meats. That's why they do that. And remember, meats and like sweets, they tend to have a very high solute or high sugar or salt environment. So that's also going to help with avoiding microbial growth. So that's done. And I can go into that further if that's not clear. Number 12, explain why gram-negative bacteria are generally more resistant to chemical biocides. We're here right now. Explain why gram-negative bacteria are generally more resistant to chemical biocides than gram-positive bacteria. Let me know if you guys found this, but the reason I found was the fact that, so remember our gram-negative bacteria, right? Pretty much all of our prokaryotic cells can be gram positive or gram negative. We're talking about gram negative right now. Remember, they have thin peptidoglycan and for the cell wall, and then it has LPS, the lipoprotein. And remember that lipoprotein is also an endotoxin. And so I think the main thing that she's going to harp on is the fact that this it, only in the gram-negative cells you're going to find that LPS, which is the endotoxin. And here's the O antigen that's associated with this negative bacteria. So gram-negative bacteria. So let me know if you found a different answer or if that's pretty much right around what you guys were thinking too. So there's that. Let's see here. 11. For each of the following chemicals, for each of the following chemical methods of microbial control, give an example of how the chemical control agents is most likely to be used. So I'll zoom in a little bit. Hopefully this will make sense. For alcohols, remember she gave the example of if you're about to get a vaccination, you have to get like a shot. So really, really hard. They'll, say this is your arm, and they'll scrub your arm really hard, really, really very hard, and they'll just try and remove germs from that area in a physical way. So that's a terrible drawing, but you get what I'm saying. So alcohols are used to degerm and remove microbes from a limited area, which would be, for example, the arm, and it's physical, and that's because it's the tissue, it'd be an antiseptic, right? So it'd be an antiseptic. Ethylene gas does sterilization, and when you sterilize, you remove all microbial life. And then you have halogens, which the two halogens that we have are iodine and chlorine. That I'm pretty sure most would agree with is that the iodine exists in a tincture to what did I say, disinfect? Let me do this. I'm going to erase this disinfect. I'm going to change this now. I'm going to change my disinfect to be antiseptic. Antiseptic. Sorry for the handwriting. I don't have my Apple pencil, so it's a little bit different. But I would say antiseptic, and the reason I change it is because when you have an antiseptic, it's trying to remove the microbial life from the tissue, and so that's why I change it to antiseptic. Because if you have a tincture that's usually soaked in alcohol, it's gonna be via a tincture, and that's probably gonna be on a type of tissue. And then you have chlorine, and for chlorine I chose disinfect because if you have chlorine, you are going to disinfect because of the fact that take for example, chlorine has the ability to um, remove pathogens. That's the definition of disinfecting. So when you're disinfecting something, you're removing the pathogens. The chlorine is a halogen, and more than likely you're going to spray like a bench or something like that, and you're going to spray chlorine on the site, and you're going to try and remove as many pathogens as you can. You're not going to use chlorine on tissue, so antiseptic wouldn't work here. So that's why I chose that. I'm sure most would agree with me, but if you don't, let me know and I can fix that. I'm going to skip this one, and I'm going to skip these two as well. And I'm going to come back to that because tomorrow I'm going to put it in much further detail. Or actually, I could probably write it here. Oh. I'm just looking right now for a second, just looking at my notes, and I'm noticing the fact that I've actually done this one already on accident. But let's see if I can use dilution test. No, I think this is a different test. So I'll just take a quick pause.